I'll introduce you because people are going to change rooms and they're expecting you to start in 4 minutes, not now.
In 4 minutes?
Yeah,
oh, okay. Good. All right. Hello, everybody. I'm Leo, and I'm a member of the iGEM team of the University of Oklahoma. And today we are going to talk about two projects which we were working on at our university. The first project was designed by Graham, here's here, and myself, and it's distributed sensor network. Oh. Well, our system consists out of two devices, as you see over here, and one device is arabinose sensor, and the other one is homoserine lactone amplifier. Well, let's go to a more detailed level of abstraction. Here you see that our system, in fact, consists out of a promoter, homoserine lactone receiver, and a homoserine lactone sensor, sender, that are connected by two reporters, which are cyan and yellow fluorescent proteins. Here is the parts that we use to construct our system, and this is how they are assembled, the order of assembly. All right, this is what we expected from our system. Well, here is a lawn of E. coli, and in the middle of it, you see it's a droplet of arabinose, and it's supposed, this area, it's supposed to turn cyan because of arabinose sensor and cyan fluorescent protein connected to it. Then the signal spreads along the lawn of the bacteria, and it gradually turns yellow. Well, so it's like kind of the yellow front kind of moves till the edge of the lawn. This is the final picture that you see, the final state of the expression. Well, and now Graham is going to talk about the mechanism of our construct. Yes.
Cool. So the interesting thing about our construct is no matter where you put the arabinose trigger, the rest of the bacteria will know that the construct has been triggered, because the cell will the homoserine lactone signal will propagate throughout the plate. Okay, now the way this works is arabinose induces the PBAD Ara-C promoter, which then turns on cyan fluorescent protein and the LuxI gene, which in turn sends out the homoserine lactone. Okay? Now, while that's going on, we've got LuxR over here listening for any homoserine lactone signal. Okay? So homoserine lactone comes in, turns on the LuxPR promoter. This in turn turns on yellow fluorescent protein, and LuxI is turned on again, acting as a sort of a signal amplifier. So as soon as the cell receives homoserine lactone, it will keep producing homoserine lactone. Okay, so this is how we ended up constructing it. We had these 6 parts from the registry, from the plate Randy sent out, and this was turned into 3 subconstructs, and then a 4th part, and then the final part was put together like so. Okay, this, I apologize for the blurriness, but this is an agarose gel of parts 1, 2, 3, the 4-parter, and then our final construct. And we couldn't actually get this construct to work properly. As you can see, 1, 2, and 3, this is Hindi 3-cut lambda here with the 2.5K and 4.5K size markers. So these are approximately the right size range, but if we look over here at the 4-part bands, which should have been 1 and 2, it's not the right size. And then if we look over here, and then if we took this 4-part and put it together with a 3-part, we end up getting 2 other bands, and we're not exactly sure what those are. So we're in the process now of sequencing these to see what part is missing and see what went wrong in the construct. So, okay, so future work, check construct via sequencing and reconstruct the part if necessary. Okay, so now Simona and Durga are going to tell about their project.
Contains their own chemical structure and properties. Small molecules play a very important role in many signaling mechanisms like extracellular quorum sensing and intracellular DNA sequencing signaling, and also in many phosphorylation events. So these small molecules are used to make drugs. Small molecule monomers like methyl methacrylate and ethylene glycol are used to make biopolymers like polyethylene glycol and polymethyl methacrylate, which are used in many tissue engineering constructs. And amino acids are used to make synthetic peptides, and DNTPs are used to make primers. Small molecule detection is necessary to understand the molecule-protein interactions and to study the effect of molecules and on the viability of the cells, that is the cytotoxicity of the small molecules on the cells, and also to understand the genetic changes caused by the molecules if they are used in molecular therapy like anti-cancer therapy or anti-thrombotic therapy. Sometimes the presence of certain small molecules in drug formulation and pre-formulation may cause the drug to become toxic. So there is a need to detect small molecules during the pre-formulation and formulation stages of the drug. Currently, NMR, Western blot, spectrophotometer, chromatography, ELISA, and many other immunoassays are available to detect the small molecules. However, we recently saw this paper which shows developed by nanowire sensors which are developed by Wang. They use these sensors to understand, to study small molecule protein interactions. But however, in the techniques like NMR, chromatography, and in nanowire sensors, you cannot understand the small molecule signaling in the whole cells. There you use just a receptor, protein receptor, or any other protein molecule, but you cannot use the whole cells. And in spectrophotometry and ELISA and in other immunoassays, you can use whole cells to understand the signaling, but the cells are static. We cannot use these techniques under flow to study the signaling under flow conditions. So in order to overcome these disadvantages, we fabricated microchannels in which whole cells can be used, and the amount of reagents which is required is very less. These microchannels can also be custom fabricated depending upon the design of the experiment, and this allows us to study under flow conditions. So this is how we fabricate microfluidic channel. This is the substrate which we use here is it can be either a glass or a silicon substrate, and we deposit a layer of photoresist, and this is a glass chrome mask which is made up of quartz, and it contains a desired channel which we want to use it for the experiment, a channel pattern. So this whole assembly is exposed to the UV light, and the place where the channel pattern is present allows the UV light to pass through the channel, and it polymerizes the photoresist present here. And after polymerizing, we remove the glass chrome mask, and we wash this glass substrate with the photodeveloper solution. So it removes all the unpolymerized photoresist present on the substrate, leaving just the polymerized photoresist, which is in the shape of the channel, and it is a raised template. So we used polydimethylsiloxane polymer and poured it onto this template, and allowed it to cure for 15 hours at 70 degrees centigrade. After that, we peeled off this template, and we got a depression in the PDMS, which we can use it as a channel. These are the channels we used for this project, and this is a straight channel which has a width of 200 microns and a depth of 50 microns, and this is a Y channel which has the angle here is 120 degrees angle bifurcation, and the channel width is 100 microns and the depth is 20 microns in the main channel. So to study signaling in these channels, we used aspartate detector to study how we can see the signaling in these channels.